Hello, my name is Andrew Dodson. I'm a member of the Chemistry 481 Synthetic Techniques lab class that participated in the Cure Transformations research project in the fall 2019 semester. Our research focused on the synthesis of polypyridyl ruthenium complexes for photodynamic therapy. Our lab prepared a series of polypyridyl ruthenium coordination complexes to support the photodynamic therapy research of Dr. Sherri McFarland. Photodynamic therapy is currently being researched for its benefits in cancer therapy. It is a minimally invasive procedure in which a photosensitive metal complex is injected into a tumor site. Upon exposure to light, a sensitizer is excited, allowing for the formation of cytotoxic singlet oxygen from ground state oxygen. The objective of this project was to synthesize ruthenium-2 complexes in order to investigate their ability to improve the process of photodynamic therapy. Four polypyridyl ruthenium complexes were synthesized, along with their respective intermediates. Each ruthenium complex is structurally similar and contains two bidentate ligands, along with an IP3T ligand. The IP3T ligand is of importance because of its ability to mediate electron transfer. Two bidentate ligands were used in these experiments. The first was phenanthrene, as can be seen on the structure to the left. The next was bipyridine, as can be seen in the list of compounds to the right. The syntheses for each of these complexes was very similar. Each was a two-step process where first a ruthenium chloride starting material was combined with two bidentate ligands to establish a framework for the ruthenium complex. Next, the IP3T ligand was attached to the remaining site around the ruthenium metal center. The products were characterized using UV-Vis spectroscopy, NMR, and cyclic voltammetry. Upon exposure to light, electron transitions occur within the ruthenium complexes. UV-Vis spectroscopy was used to determine at which wavelengths these transitions occur. UV-Vis data was collected for the ruthenium complex intermediates and the final ruthenium complex, including the IP3T ligand. Seen in both the intermediate complex and the final complex is a sharp peak in between 260 nanometers and 275 nanometers. This peak corresponds to an intraligand pi-to-pi star transition between the two bidentate ligands. Seen only in the IP3T containing ruthenium complex is a broad peak near 420 nanometers. This peak corresponds to metal ligand charge transfer transitions between the ruthenium-2 complex and the bidentate ligands. NMR analysis was used to confirm the structure of the IP3T ligand and each ruthenium complex. Proton NMR and cozy NMR were used for such analyses. The hydrogen atoms of IP3T are labeled A through J and correspond to chemical shifts on the proton NMR spectrum. Similarly, the hydrogen atoms of the ruthenium complex are labeled A through N and correspond to chemical shifts seen in the proton NMR spectra and the cozy NMR spectra. Upon attachment of the IP3T ligands, there is a downfield shift of IP3T ligand protons. The proton NMR spectra confirm the expected IP3T ligand protons and metal complex protons. The cozy NMR spectra also confirms this. Our research project concluded with the synthesis of four ruthenium complexes, each containing the IP3T ligand. The IP3T derivatized ruthenium complexes were shown to exhibit enhanced light absorption properties in the visible region. The structure of each ruthenium complex was confirmed using NMR spectroscopy. The synthesized compounds have been sent to Dr. Sherri McFarland at UT Arlington for biological testing. On behalf of myself and the rest of the Chemistry 481 Synthetic Techniques lab class, I would like to thank Dr. Sherri McFarland for allowing us to participate and contribute to her research. I would also like to thank the Cure Transformations Council of Undergraduate Research and the UNCG Department of Chemistry and Biochemistry. I would especially like to thank Dr. Jerry Walsh and Abu Stoyev for their help and guidance during the project.